Brian Gratwicke. And but first, I'd like to give you one thing to keep in mind as you're listening to everything that he's saying tonight. And that is a very sad statement about where we are. Amphibians are the most threatened class of animals on the planet. And there's a lot of reasons why that is important, not just to them and the threats that they face, but to humanity. So he will get into that. And our speaker, Brian, is one of the worldwide leaders on the issue of amphibian research. He's a conservation biologist, and he leads the amphibian conservation program at the Smithsonian's National Zoo and Conservation Biology Institute in Front Royal. And we're really thrilled to have you here tonight, Brian. His special focus is on building capacity to conserve amphibians in Appalachia and Panama. And fascinatingly, and we'll hear more about this, he leads a facility in Panama to maintain and grow what is called assurance populations. And he'll explain that, of the most threatened species. And closer to home, he also conducts research into emerging diseases and the effects of climate change on Appalachian salamanders. And with that, Brian, take it away.
Thank you so much. When I was a kid, I used to cycle past zebras and giraffes on my way to school, dreaming of salamanders. And there were no salamanders in Zimbabwe. The salamander capital of the world is right here in your backyard. Wow. And this is one of the things that makes the United States a unique continent, is its salamander biodiversity. It sets the United States apart from all other places in the world. You know, if you think about bald eagles, national symbol, we got fish eagles in Zimbabwe, they're basically the same for that, right? But there's no salamanders, there's no tailed amphibians in Africa or Australia. So this is one of the things that just really captivated me as a kid. And so when I flipped over my first log in Rock Creek and found a redback salamander, I was ecstatic. You know, it hits all of the right little notes, and it just gets all of those little sparks going in my brain. So the thing is, I started off as a fish biologist. And when I came to the United States, there was this sort of bubbling concern about amphibians. And at the time, it was in the year 2000, the status of the world's amphibians had just been figured out. And
yeah, just left click.
Okay, the status of the world's amphibians had just been released, and it wasn't good. And we've just repeated that same analysis 20 years later, and the situation is dire. About 40% of all the world's amphibians are threatened or endangered. You compare that to other groups, other highly threatened groups include cycads and conifers and corals, but you compare this to birds, and just 13% of birds are threatened or endangered. You know, but if you add up the top bird conservation organizations, they have about 8,000 full-time employees working on bird conservation. More than $1 billion are being spent annually on bird conservation, whereas in amphibian conservation, there's really not a lot of us out there doing this full-time. There are a lot of academics that study amphibians as herpetologists in universities, but there's not a lot of people out there taking actions to mitigate threats, right? And so what are the threats to amphibians? Well, diseases, climate change, habitat modification are the really big ones. And when you look at the map of where you find most of the threatened amphibians in the world, you know, you get some real hot spots, and particularly in the Andean region and up where I work, mostly in Panama, very, very dire situation. We have about 200 species of amphibians in Panama. 9 of those species are probably extinct. Some of them exist only in captivity in our amphibian rescue center in Panama. But up here in the United States, not so much not so much of a hotspot for threatened amphibian species as in the neotropics. But we do have a really interesting concentration of salamanders. If you compare frogs, Virginia has about 27 species of frogs, and this is about 0.5% of the world's total frog species. But Virginia has 7% of the world's salamander species, just living in Virginia. 52 salamanders, twice as many salamanders as we have frogs. This is what makes this place special. Look at that map. We're saying that this is what distinguishes North America as a continent, is its salamanders. It's not its bald eagle, it's its salamanders. And they're pretty awesome creatures. These are the tailed amphibians. These are the ones that do not lose their tails when they metamorphose. Frogs, the tadpoles lose their tails, they get reabsorbed back into the body, and then they are more adapted for terrestrial life. But these creatures are pretty amazing and diverse and colorful and slimy and just So I've spent a lot of time noodling around logs and looking at salamanders up in the park and all over the place. I tell you what, I've been working in Panama now 15 years, and I flipped a lot of logs in Panama, and I've only ever seen one salamander there. And this is it. It's the Colombian worm salamander. There are some diversity of salamanders down there, but as our project director in Panama, Dr. Roberto Benes always says, they will find you. You don't find them. So they're just these really rare chance encounters. But you can go out on SCBI property and flip a few rocks and logs, and you'll see 7 different species and hundreds of salamanders in a morning. So it is very special. And this is the first salamander I ever saw, apart from an axolotl that I had in my bedroom growing up. But this is a redback salamander, and it's a very, very handsome fellow. And I'm going to make the case as to why this is the coolest salamander ever. These are one of the most abundant animals in the eastern United States, period. One of the most abundant vertebrates. If you were to take, even at mean densities of redback salamanders within their range, you were to take all of them in the forest and put them on a scale and take all the deer at the highest deer densities. Think of the place with the most deer ever. Put those on the scale next to just a medium density of redback salamanders. And the redback salamanders would probably break about even with the deer, right? If you go to a high density area for redback salamanders, they'll tip the scale in favor of the salamanders every time. The only other real vertebrate that achieved this level of biomass in eastern United States was the passenger pigeon, and it's extinct. But if you were to look at bears don't even come close to where you have the lowest density of redback salamanders. So this is one of the cool things. I contributed to this research, counting lots and lots of salamanders at our property at the National Zoo and Conservation Biology Institute in Front Royal. So where do you find these lungless salamanders? Well, they're lungless, so they need it to be cool and moist. They do not like it hot. When it gets too hot, they go underground. When it gets too cold, they go underground. When it's just a nice day, they go underground. So they don't spend a lot of time on the surface unless it's about 15 Celsius or about 60 degrees Fahrenheit. That's like their Goldilocks temperature, but it also has to be moist. So 100% humidity and about 60 Fahrenheit. So this data I collected from our study of salamanders out of Front Royal, and it just goes to show, hey, you can go and count all of these salamanders in the forest, but how do you know how many you didn't see?
Yeah, right.
Right? Well, we have an array. This is called a SparkNet plot, and this is our George Mason University, Smithsonian Mason School of Conservation out at SCBI. We have these cover boards. They're just little planks of wood, and the redback salamanders like hanging out underneath these cover boards because it's nice and protected and it's moist and just right for salamanders. And we go through with the students and we gather up all of the salamanders under these boards when we find the most salamanders when it's about 60 Fahrenheit and 100% humidity. And then we give them a little tattoo that fluoresces when you shine a black light on it. And we give them this unique tattoo so that we know who's who when we catch them again, right? And then through the process of marker capture, it's a simple mathematical ratios. We look at the ratio of marked animals to the ratio of unmarked animals. And by doing a little math, you can calculate how many salamanders you didn't see. And after doing that, we found that there were about 12 to 16 million redback salamanders on SCBI property alone. And why are they the coolest salamanders? Well, the third grade at Powell Elementary School in DC thought that redback salamanders were the coolest salamanders in the world because in the pandemic, those students would go walking in Rock Creek and they would flip rocks and logs and they found redback salamanders there. And they said, you know what? DC does not have its own state amphibian. So this group of students went and lobbied the DC City Council and said, we want redback salamanders to be the state amphibian for DC. And they called me up and they said, Dr. Gratwicke, what can you tell us about redback salamanders? I said, I can tell you lots of things about redback salamanders because they are one of the coolest amphibians in the world. They're the most abundant. They're salamanders. DC is the capital of the United States, but this place is also the capital of the salamander capital of the world. And this is one of the things that makes and stars and bars. It's a redback salamander. What more could you want? We made these T-shirts. So when they went their council members, they were ultimately successful. And this is now the state amphibian for DC. And those are the kids that did it. And it was made quite a lot of headlines. So salamanders are very cool. Redback salamanders are amazing. And a lot of people will call me up and they'll say, Brian, I just saw a Shenandoah salamander in my backyard. And I'm like, no, you didn't. It's your backyard, the national park. And they're like, no. It looks, I'm looking at a picture of the Shenandoah salamander. I'm like, yep, they look pretty much like the same thing. But they couldn't be more different in conservation terms. The Shenandoah salamander only occurs in 800 acres anywhere on the planet. Whereas the redback salamander is one of the most wide-ranging, abundant vertebrate in eastern forests. And they are closely related, but they are distinct species. So where do you find Shenandoah salamanders? Only on north-facing talus slopes inside the Shenandoah National Park. So they are like our most endangered amphibian in Rappahannock, and they are pretty cool. And you can find them in spring and fall, even on the Appalachian Trail in some places. And when you look at them together, they're still kind of hard to tell apart. And this one here is the Shenandoah salamander, and this is a redback salamander. So You might not even know that you saw a Shenandoah salamander if you did see one in the right place. But the key is when you flip them over, the Shenandoah salamander has this dark belly. But a lot of us do not have the national park in our backyards. This is what we have in our backyards. We have a pond and, you know, maybe we have some wet squishy areas where frogs like to breed and that kind of thing. So when we moved to Rappahannock, we inherited this beautiful pond and it's full of bass and bluegills and bullfrogs and it's pretty cool. But where do you find these cool critters? These are some of the cool critters that you find in fishless vernal pools in this region. And when you have a fishless vernal pool, it's something very special. It's a wetland that these animals can come. It's not flowing water, it's still standing water. So it has a lot of zooplankton and good stuff for these amphibian larvae to eat. If it's clear, gentle, flowing water, there's no plankton. It needs to be still water for all of these plankton to build up that these tadpoles, these larvae of these animals can eat. But if there's fish in there, the fish will just gobble up all the tadpoles and it's game over. So a lot of these amphibians will absolutely not even lay their eggs in a pool where there are fish. So if you give them a little bit of water without fish, these animals will come and they are some of the special animals. And I think they're the special animals because a lot of people, when they get these wet, squishy areas, they're like, oh, that's a little swamp. That's something that needs to be drained. We don't want mosquitoes. And they tile it, they'll ditch it, they'll get that water off the surface. And that's been happening all over. Even our SCBI campus, we have lost about 1/3 of our first order streams that normally would be habitats for these kinds of amphibians. So these guys have not been doing well, but these guys have been doing very well because there's been a lot of farm ponds that are stocked with bass and bluegills. And these guys can handle the fish, right? Bullfrogs, green frogs, red-spotted newts, they have tetrodotoxins. They're quite toxic. They are also voracious predators of other tadpoles. Pectoral frogs, they can all hang out with these fishes. Now these amphibians, while I love them and they are native, they're definitely already facilitated on the landscape, right? The first group has really been hard done by in the current habitat of Rappahannock. This group is sitting high on the hog. They are getting subsidies all over the map, right? And the other ones are getting no subsidies at all. But when you do build some of these, maybe you'll create a fishless habitat and say, okay, this is great. It's a fishless habitat, and we're going to make this we want those spotted salamanders and all of the cool vernal pool breeding amphibians, right? But guess what? Those previous groups that I showed you, the bullfrogs, the green frogs, the pickle frogs, they can live with fish. They can live better with fish when there's riparian vegetation, but they'll quite happily live in a fishless pool. They'll be very happy to be in a fishless pool. But these guys can kind of persist over time. And so this bullfrog tadpole is 3 years old. This is what happens when your fishless habitat does not dry up. You get absolute chunkers of tadpoles that basically are fish, right? And so now you've created a nice fishless habitat, and because it hasn't dried up from season to season, it's actually a fish habitat inhabited by amphibians that have turned into fish because they've just taken 3 years to metamorphose and they get really big. So this is why it's important for these temporary habitats to dry up from time to time, to clear the green frog and bullfrog tadpoles out. And those red-spotted newts too, They love to eat other amphibians. So we just did a project with one of my master's students at George Mason University. This is Jason Motley's work. And he worked with the Virginia Master Naturalist Vernal Pool Monitoring Network and took all of their data and said, okay, let's see what we can analyze and learn from this. And this is the phenology of these salamanders using vernal pools monitored in this network. And what you see is Jefferson salamanders occurred in about 8% of the vernal pools. And the eggs are quite distinctive, and so those are very laid very early. You'll see the first ones in January and February, right? So they're these early into the water breeders, some of the first in the season. Why are you going to do that if you're a vernal pool breeder? Because you want to be there first before there's other competition with other tadpoles that might eat you or compete for food, right? And then the spotted salamanders come in just a little bit, just a shade after that, and wood frogs about the same time, early. Marbled salamanders, you'll see actually their egg masses are normally laid around about now. So they are very early into the water. But the rarest one is the Jefferson salamanders. 60% of the vernal pools had spotted salamanders, 31% of them had wood frogs, and about 30% of them had marbled salamanders. So when you're looking at, hey, what's a really, this is a habitat that's not doing well. A lot of people have drained it. A lot of these animals that should be in high abundance on the landscape are no longer in high abundance. But Jefferson salamanders, this is an interesting creature. They are actually related to the blue spotted salamanders from further north. If we were further north, these would be blue spotted salamanders. And these are very interesting animals. And when we plotted out the distribution of Jefferson salamanders and just colored in the dots from the last 10 years in blue and the historical dots from any time before the last 10 years in yellow, we find that their range has contracted significantly. The conservative estimate is they've lost about 40% of their range in 10 years. The maximum sort of estimate, if you actually extend those to all of those yellow dots on the map and all of the blue dots on the map, that's like a 62% range contraction. So this species it's rare, it's not doing great, and seems to have a stronghold in Virginia. This is the one place where it is still doing okay, even though it's only at 8% of the sites. When we did a habitat suitability mapping for this species, this is the red colored areas are the most suitable places for Jefferson salamanders. And you can see we're somewhere around about here. So we're kind of right on the edge. There's a population at Clifton, but it's an interesting rare animal, and I think it is worth paying some attention to. But we do not. I know a lot of you are like, okay, well, how can I help Jefferson salamanders? I'm going to move some from a nice spot where I could found a pool to my property and give them a new home. They could probably live here, but Virginia, the DWR is very much, we do not move amphibians on the landscape because you can move unintentionally amphibian diseases, right? I took Bruce out to one of our vernal pools on SCBI property to see some wood frogs, tadpoles, and we came along, and what did we see? We saw a whole load of dying wood frog tadpoles, and it was because of a rhinovirus infection. So it's not just the amphibian chytrid fungus that I'm going to touch on a little bit later, but rhinovirus in this part of the world can be spread. So we don't want to be moving amphibians around the landscape, but very much if you build it, they will come. They will find it. At Dick's house, he dug a little vernal pool situation, and literally the next day it was full of eggs.
Yeah.
So it can be very, it can be very rewarding. So I think one thing I want to touch on a little bit now is soundscapes, because I've just gotten into this area of research and I have found it to be very rewarding. This is the iNaturalist website. If you are not familiar with it, you should have iNaturalist on your phone, and you can submit sounds on iNaturalist. So you can go out, you can record an owl or a frog calling, and you just click the little record button and you submit it, and people will help you identify what that sound is. But now there's 120,000 frog calls sitting on iNaturalist from about 1,700 species. That's an amazing collection of sounds. And so we have these sounds and we can use them as vouchers of the presence of an animal, right? And I hope this one works. This is not as impressive as I was hoping for, but that is the trill of an American toad. And I'm super glad I didn't spend like 5 hours making lots and lots of these videos today. But it's one of the sounds of spring, right?
Yeah.
And it's the soundscape. And when you put in these little vernal pools, you restore the soundscape, right? You are restoring the sound of nature. Nature's soundtrack is spring amphibians. And as an amphibian researcher, we can now use artificial intelligence to build classifier models and train it to recognize these sounds. Just like the Merlin app for birds, we can do that same thing for amphibians. And we can deploy these acoustic monitors into the environment, and they'll record whatever duration you want. I do 1 minute every 10 minutes, and we can monitor the calls and the presence of amphibians on the landscape. And then we can feed it into these pattern matching analysis. It's like Shazam. You can just say, show me anything in this collection of 100,000 calls that would take 50 days to listen to. Just show me the bits that sound something like this. And it'll look at the frequency and the timing and the pattern inside that bounding box and go and look for things that look like that, right? So it's pretty cool, and it has been very useful to me in Panama, where I work. You know, one of the most impacted areas by the amphibian chytrid fungus. A lot of frogs have disappeared in that landscape. And you can put out these sound recorders, and this is what the different calls of all of these different species up here look like. And what we have found is that a lot of these species we thought were extinct, we've now managed to find through this calls on the landscape, which is pretty cool. There's still a whole bunch that are missing, but it's been very, very rewarding to actually rediscover some of these species and then go out and actually, these photos are taken of the actual ones that we rediscovered through the acoustic monitoring. And then I have a brilliant conservation biologist, Nate Weisenbeck, who's very good at finding these creatures, and he just sniffs them out. And it's been really, really amazing to see this happen. So we are starting to find some of these lost frogs. But I want to tell you something, like I'm starting to put some of these. That's a little louder, that's better. That's the sound of spring. Now, spring peepers have the peep, but they also have this little male aggression call that's kind of like a little trill. And it also sounds to me a little bit like someone running their finger over the teeth of a comb, just just like a little trill. And I was listening to my little vernal pool and thinking, you know, could they be chorus frogs? Could there be chorus frogs inside the chorus making that little trill sound? And that's what a chorus frog sounds like. And they apparently occur in Rappahannock. I just never seen one. So I was kind of hoping that deploying some of these acoustic monitors might help us pin down some of these chorus frogs. Because if you've ever tried to see a spring peeper, even though they're calling and they can be very loud, it's very hard to actually lay your eyes on one. And I'm just wondering how many other Pseudacris, they're the same genus, how many other Pseudacris are in that chorus? And we're just not picking them up because we're not familiar enough with the call. I've never seen one in Rappahannock. I would very much like to. But I think that there are some of these more sort of cryptic opportunities for some of the sound work around here. So I'm very excited about that. But you may have seen the link that was sent out with this announcement. The Washington Post came with me to Panama and covered our frog project there. And we were doing a lot of release trials. Right now we have 4 active release trials in Panama with different species going from our captive bred populations back into the wild. And this article was pursuing the relationship between frog declines and upticks in malaria in the human population, and covering our efforts to try and recover some of the frog populations in Panama. And this was one of the stars of the article. And I'm going to give you a little update on that, because this is some of the work that Nate Weisenbeck has been doing. And this is Pratt's poison dart frog. So it disappeared from inside a national park, inside the Campana National Park. And it had recovered. We discovered through our acoustic monitoring that it had recovered at another place, about 60 kilometers away. So we went and managed to get 12 pairs, male-female pairs of these frogs, and stick them in little cat play tents inside the rainforest, inside Campana National Park. So here's a little map of where we have little audio moths here, and each of these black dots is a cat play tent that had a pair of these frogs released into it. And what happens when we did the acoustic monitoring, this is clearly before we put the frogs into the rainforest. There were no calls of this species. When we put the frogs in, suddenly there were calls. And then this pink arrow is when we opened up the little cat play tents and let the frogs out into the world, right? Mm-hmm. And so what happened is at these sites, the frogs persisted and they called over time, right? And at this other site down here, it looks like when we released the frogs from the play tent, a bunch of them came from up here down towards the stream. So that's pretty cool to observe. And then up here, we started hearing some of these frogs a little before we released most of the frogs. And what happened there is one of the frogs had escaped from a tent early. So he had released early. And over here, we had some transient frogs coming through a little later. So we can actually monitor the dispersal across the landscape with this pattern matching approach.
What's the time frame that you have on that bottom axis?
How do I do this?
You want to go back?
Here, so each of these is a month. June, July, August, September. So that's 4 months. That's a 4-month period. And what we're finding is that these poison dart frogs are persisting very well. Just last month, Nate went and laid his hands on 7 of the males and actually swabbed them. And so we can see that they've been dispersing outside of the area where we have been monitoring. So there's good reason to believe that this is going to be one of the first successful conservation mitigations of the chytrid fungus in Panama. I'm very hopeful that's what this will be. But we've also been releasing a lot of other amphibians, and probably the most rewarding moment for me was this. We got to release Panamanian golden frogs back to where they should be, out in nature. And I have walked these streams for 15 years, imagining one of nature's missing puzzle pieces back in its place. And I finally got to see it. And it was probably one of the highlights of my career. But I want you to think about that a little, because when we have built this amphibian ark in Panama for 9 different species of frogs, it has been extremely difficult, extremely expensive. We grow every cricket that goes into these frogs. We clean all the frog food. We look after these animals. We struggle to reproduce them, to produce enough animals for research to understand the chytrid fungus, to find a solution, to find a cure for the chytrid fungus, so that we can recreate nature. And here, and my husband dug me a vernal pool for our anniversary. And, he's like, well, why are you so into all these vernal pool things? And I said, well, these are little amphibian factories. And I don't even have to raise the crickets. All you have to do is just dig a hole and let nature do the rest. And it's so rewarding to just see how responsive it is to just have some of these small little actions that you can take to weave a place in nature's fabric for all of the participants. So that is what I came to pitch you tonight. And with that, I'll take questions.
The frogs are releasing in Panama, do they have some immunity to the fungus, or how are we?
Well, the Pratt's poison dart frog, we believe, have evolved immunity to the chytrid fungus. Some of the others, we have tried different mitigation tricks to put our thumb on the scale in favor of the frog. With one species, we have actually put antifungals inside little tadpole mesocosms where the tadpoles can grow, to give them a little boost into the world. There's another species of frog, the crown tree frog. We've created lots of additional breeding habitats, so that if lack of breeding habitat is a limiting factor for the population, maybe we can boost the population by facilitating that. And even though some of them may die from chytrid fungus. For the Panamanian golden frog, this particular research, they are still susceptible. About 70% of the frogs have died from the chytrid fungus. But this is rewilding research, and our goal is not to create a new population of Panamanian golden frogs back in place. It's to understand how do they regain their skin toxins so that they can ward off predators. I have a research program that is looking at potential climatic refuges from the fungus. And in some of those places where we still find harlequin frogs in Panama, seem to be unsuitable for the chytrid fungus about 80% of the seasons in the last 15 years. So if we can select the places where we put these back, they may be able to persist, maybe not thrive, but they may be able to persist in some of these places. So that's the research. And it's one foot in front of the next. It's just a lot of hard work and discipline.
Brian, this is Mike. Could you repeat the questions for those of us that are on Zoom and for the recording too when somebody asks one?
Sure.
Thank you.
Yeah, Rappahannock County, you probably know that we've had long periods of drought over the last couple of years, couple months blocks at a time. Have you seen any noticeable impact on the salamander population or amphibians in general?
The question was, is there any impact on the amphibians from the droughts? The last drought we had, I had all of the salamander tadpoles in my vernal pools, and they were drying up, and they were drying up too early, and so I got buckets, and it was a bucket brigade. So my ones have been able to survive drought. But it is part of nature's cycle. We get, these vernal pools are supposed to dry up. These are explosive breeders. When you have a successful vernal pool and you walk after the toads start coming out of the water, you can't even find a place to put your foot without treading on a frog, because the ground is covered with them. So when you have a good year, that should balance the years that are not good. Salamanders, these spotted salamanders, can live for about 30 years. In fact, some of the salamanders on the landscape, if you built a vernal pool, may have been looking for that vernal pool for the last 15 years and not been able to find one. So they will come, they will find it. But if you go too long without an opportunity to breed, it's not going to happen. Yes.
So this is a wonderful talk. Thank you very much for educating us and also what you're doing. I'd like to know if you have 3 recommendations of things that Rappahannock residents can do to try to encourage salamander and other amphibian growth. Sounds like a vernal pool is on the list, but what else would you recommend?
So the question was 3 recommendations to facilitate amphibians on your property. I would say number 1 is a fishless habitat that dries up from time to time. Would be great. It can be a vernal pool, it can be a wet squishy area, you just dig a hole, or it can be a lined pond, just like you can go to Lowe's and buy one of these ponds. Nancy has a wonderful pond that does not have any fish in it, but gets frogs every year, and it's one of these Lowe's plastic ponds. It's great. So there are, if you build it, they will come. If you have a pond with fish in it, you could consider a refurb, which is if you get the pond people to come and drain it and maintain it and do what they do to older ponds. When they fill it again, you can just decide not to put fish in it. That would be great. If you like your fish, you can have fish. You can make it better habitat for the existing animals like the bullfrogs and the leopard frogs. And you can have a softer edge. Native plants, trees, softer edge really will provide cover for all of these animals. So if you have a pond with fish in it, let's soften those edges. Let's get some native aquatic plants on the margins. And then if you have woodland that is older woodland, chances are it's going to be good for redback salamanders. But they really need old woodland. So our property has maybe a stand of 20-year-old tulip poplars. I've seen 2 redback salamanders on our property the entire time I've lived there. So they're not, they're still suffering from the time when it was cleared out, right? So if you have some older growth woodland on your property, be a good steward of that, and think about some of the creatures that are the highest biomass of any vertebrate. Yes.
If they're laying their eggs in December and January, what keeps them from freezing?
Sometimes they do freeze, and sometimes if they lay their eggs in January and February, what keeps them from freezing? So with the current seasons and climate warming, you know, we had a 70-degree day in January that pulled out box turtles 2 years ago. And I was like, this is, and then they died from the subsequent freeze, right? So if you mess with their annual cycle too much, you can have animals come out too early and they jump the gun. Normally, a little freeze is not going to hurt any of these pond breeders. The spotted salamanders, wood frogs, they're going to do just fine. But if it freezes if it's really cold and it freezes all the way to the bottom, they will die. And so you don't want them to be jumping the gun. So you really want the seasons to be more historical than the current warming scenario, because this warming scenario wakes everyone up and gets them out of bed too early and then zap. So we just, you know, it is definitely a thing, and I think there are some people starting to study this, the sort of the timing of these emergencies. And one of the greatest resources for this is iNaturalists, because a lot of people will go out and say, oh, I've seen my first wood frog egg mass, and they'll take a photo of it. And you know what? It's a voucher. It's a photograph of an organism at a time and a place. And if you have that voucher, you can do all kinds of great research, like research into climate change and all the kinds of things. So it's pretty cool.
Brian, what are we talking about in terms of size of a vernal pool? When was it go out and dig one? How big does it need to be? Is it?
You should ask my husband. I like them to be as big as possible, but you know, about 10 feet by 5 feet wide.
That'll do it.
10 by 10 is better than 10 by 5, but you know, 10 by 5 is a good size. You'll be well rewarded with a vernal pool of that size.
And are there other conditions that a vernal pool should meet? Sun direction, or any other conditions you should be aware of?
For things like spotted salamanders and wood frogs, should be near a woodland, because they're going to be moving out into the woods and then coming to this breeding spot.
Okay.
For things like tree frogs as well, some vegetation nearby is helpful. If it's out in the middle of a field, things will still find it and they will still use it. Spring peepers and everything will still just love it.
Mm-hmm.
Brian, what would be the ideal depth of a vernal pool? Is there an ideal situation?
The question is,
be it either woodlands or an open field.
The question is, what is the ideal depth of a vernal pool? I tend to keep them about knee deep. That's sort of my preferred one, because then it gives them a chance to dry out. If they get a lot deeper, then the bullfrogs and green frogs will move in. It doesn't preclude everything else from working there, but you know, I tend to try to sort of put my thumb on the scale in favor of some of the more disadvantaged amphibians from around here, because I think.
And an aside, you were very fortunate to come out to our place. I was so excited last mid-March, and told you, Brian, that I counted 17 or 18 salamander egg masses. And of course, you were excited too. And then you came out 3 weeks later with your trusty net and netted for 15 or 20 minutes. And how many salamanders did we find? 0.
I'm going to repeat the story for our Zoomers. Bruce has a little pond that is not exactly vernal because it doesn't dry up.
Right.
And he got 18 salamander egg masses one spring and said, I want to see the salamanders. So I brought my net out and we netted for the salamanders. All we found was bullfrog tadpoles, no salamanders. And that's probably because the bullfrog tadpoles were hungry. So it is important to just pay attention to the ecology, to the phenology, to the environment.
So Brian, thank you for helping me locate a vernal pool, but it's not drying out. How often should you try to have it dry out? Like I could channel it or something. Every 2 to 3 years, every year? What's the best practice for that?
Best practice should be dry out every year, but it's nature. These are just small fishless wetlands. So a lot of them just have their own rhythm, right? And they will provide a space for amphibians in that. And if yours doesn't dry out, I wouldn't fret about it. I would just see what uses it and just pay attention, just be observant.
Brian, those acoustic boxes that you were talking about, they can be attached to a tree and then can recognize all the different kinds of.
They cannot recognize anything yet. We have the amphibian. Sorry, the question was the audio moths. Can they recognize all the frogs? And the answer is no. They can't recognize any frogs because the bird people, as I said right at the beginning, have 8,000 full-time employees spending $1 billion a year. The frog people are spending a lot less than that. And I think we could build out the Sounds of Nature project. We could make like the Sounds of Merlin, price of $2 million with Scott Laureate iNaturalist. I think we could do that. I think we could build it. But it needs a lot of labor, it needs a lot of volunteers, and more importantly, it needs some of the infrastructure, the back-end infrastructure to do the sound annotation, because the whole point of AI is you've got to actually teach the machine first. And the way you teach the machine is you've got to annotate a lot of data and then say, this is this and that is that. And that's what we need to build. They've built it for birds. The Cornell Lab of Ornithology has done a great job. They've got 2 different flavors. They've got the Merlin, which is done through eBird, and then they have another one that is attached to a thing called BirdNet, and the BirdNet models inform the haiku boxes that Dick has shown me. Like you can leave them recording all day long, and you can come home and you'll say, well, what did we have today? And it'll say, you heard 32,000 house wrens. So yeah, we're not quite there. We're not anywhere even close for frogs, but I can see us getting there. I'm excited by it. I can see the value of it to conservation, very applied values in all kinds of ways. So we are starting to do some of this kind of the hard way, and building out the models one by one. So
Can we see if there are any questions from the Zoom?
Yeah.
Mike? No questions?
Yeah, this is Mike. I'm sorry. I'll turn my face on. I'm not sure where I am here. No, I'm sorry. We've got no questions from Zoom, but I'll remind everybody that's on Zoom that if you've got one, put it in the chat box.
Any other questions in the room?
I have questions about going back to the first map about the geographical distribution. I'm noticing there, for example, with you talking about the red salamander, for example, being heavily in parts of the United States. I noticed the Appalachians have some, the Rockies don't seem to have any. Is this all a matter of moisture, cold temperature? I mean, is there a variable in there that is determined? I mean,
the Appalachian. So the question is, why the eastern United States why the geography of the distribution of of salamander biodiversity hotspots. Why is it concentrated in Appalachia? Well, these are the oldest mountains. These are the oldest mountains. And we've had a lot of sort of climatic changes with the ice ages and back and forth. And this creates new sort of disruptions and isolations of these amphibian fauna. And it leads those periodic isolations leads to the evolution of new species, and particularly on mountaintops. If you look at the Shenandoah salamander, it's a mountaintop endemic. There's also the cow knob salamander. There's also the cheat mountain salamander. There's a lots of these mountaintop endemics, and they only exist on these sort of what we call sky islands. And we're worried with climate change that with raising temperatures, they'll just be forced off the top of the mountains. There'll just be nowhere more for them to live. But historically, if an ice age comes through, they can suddenly occupy a lot lower elevations, and there'll be movement over millennia, and that leads to the process of separating these organisms for a long enough period of time that it'll allow genetic drift and natural selection to change these species so that you end up with lots and lots of different species.
To the salamanders, I've been wondering whether the decrease in insect populations was affecting them.
The question is, what do salamanders eat, and has the decrease in insect populations affected them? The little woodland salamanders like to eat a lot of the leaf litter invertebrates that are decomposing things, roly-polies and springtails and those sorts of things. The pond-dwelling amphibians, they will, as adults, they will eat a lot of the animals that like to eat your garden, so they will keep the pests down. And as tadpoles, they'll eat mosquito larvae. So they really do eat a lot of different things depending on what species they are and what life stage they are. Some of the tadpoles will eat more algae. Some of them really are very cannibalistic. Some of these spotted salamanders are so cannibalistic they'll just eat each other when they run out of food. So there is quite a variety of diet, and bullfrogs, these big bullfrogs, they'll eat a small bird if they can catch it, right? So just about anything that moves.
What's the best time to construct new vernal?
Every day of the week. But funnily enough, now is the best time. Now, particularly in fall, I think it's a good time because it's a little before the spring breeding. You're not going to disrupt anything, and then come spring, you should get rewarded. Okay, thank you.